Okay, good morning, everyone. Anand, please.
Good morning, everybody. This morning, we're going to do an update on the therapeutics around COVID-19, or how COVID trials are changing the face of clinical research in Manitoba and around the world. We have two very distinguished speakers with us. Dr. Ryan Zarychanski is an associate professor, critical care medicine, hematologist, and clinician scientist at the University of Manitoba and Cancer Care Manitoba. He is, as many of you know, a senior scientist at the Research Institute of Oncology and Hematology, maybe you don't know, and an affiliate member of the George and Fay Yee Center for Healthcare Innovation. What you may know is that his research focuses on the hematologic aspects of critical illness, where he leads several national and international randomized trials in the field of sepsis and transfusion medicine. Dr. Zarychanski leads the COVID-19 clinical trials team in Manitoba and is the chair or co-chair of 3 international trials evaluating anticoagulation strategies and convalescent plasma in COVID-19. Sylvain Lother is a critical care and infectious diseases physician at the University of Manitoba. He is one of 3 of us, along with Dr. Light. He's currently completing a Master's of Science in the Department of Community Health Sciences and a clinical research fellowship under Dr. Ryan Zarychanski and Yoav Keenan, who's also here. Gaining practice experience in the conduct of randomized trials, Dr. Lother contributes leadership to the ward-based COVID-19 trials in Winnipeg and is involved in several of the international trials led out of the province. Dr. Zarychanski.
Thank you, Dr. Kumar. Thanks for everyone for joining today. On the Zoom part of the call is Yoav and Anand and John Amble to help answer questions at the end of the presentation. Really pleased to do this with a very senior trainee and physician, Sylvain, who will be presenting many of the slides. All right, let's get into it. Okay, so if you have questions to submit for the presentation, click that link. If you have received the post already, I'll take you right to the place. But if you don't have this clickable link, then go to slido.com, and then there's a code, enter hashtag medicine. I don't know if the capital M is important or not. And then it'll direct you right to the place where you can ask questions online that we will field at the end of the presentation. Okay, some disclosures. These are grants related to COVID-19 research and for the randomized trials. We do national grants, provincial grants, foundation grants, and some from Ontario as well, where we have co-PIs involved in our trials. We're going to provide an update. That's the major point of this, is an update on therapeutics as we see a surge in Manitoba and make beds available in different severity of care areas. How will we care for these people, and what's the evidence-based strategies that we can provide? We'll talk about Manitoba's clinical trial response to COVID-19 in the context of a learning health system, which is also very new for Manitoba, but something that I think that will be enduring and that will eventually move to as a province over time, and highlight how investigators in Manitoba are leading some of these trials and how it increases our research capacity, puts us on a different stage where we may not have been before. Okay, I'm going to turn over a lot of it to Sylvain, and then we'll go back and forth. So Dr. Lother.
Thanks very much, Dr. Zarychanski. So as you can see here from the Canadian epi curve, this pandemic continues to be a huge problem in Canada. And while we were able to control the first wave with public health interventions and shutdowns, we're facing a second major surge, and it remains to be seen whether our current mitigation strategies are working. Over on the next slide, this time around, it's very evident that this is becoming a huge problem in our own backyard, which is different than the first curve we saw, or the first wave we saw in Canada. This is happening right now as we experience a massive spike in community transmission. This graph here illustrates the daily case count per million residents. And you can see here that rates are skyrocketing past the majority of the US and even our neighboring provinces that have struggled earlier in the pandemic, like Quebec. If you go over to the next slide there, Ryan, Canadian outcomes, why is this a problem that we're seeing such rise in community cases? Well, Canadian data tells us that approximately 8% of our community cases will require hospital admission, with a high need for ICU treatments and in-hospital mortality. The ICU admission rates is approximately 1.7% of all community cases, many of which need ventilators for long periods of time with high mortality and risks increasing up to around 31% mortality on the ventilator. So to give you a sense of what that will mean for our hospitals, we can calculate, if you assume that we're having about 250 community cases per day, we're actually seeing a lot higher than that lately. Soon this will translate to approximately 20 daily hospitalizations and 3 to 4 daily ICU admissions. These are numbers that will very quickly outstrip our resources. So if we go over to the next slide, the spectrum of illness, most of those hospitalizations will come in to hospital around day 5 to 8 of symptom onset, which is kind of what we've been seeing. So if we think about the pathophysiology along the spectrum of illness, we will be catching the virus sort of at the tail end of its replication phase and dealing with symptoms that are more likely related to the host response, a principle that will frame our approach to studying treatments and various treatment options.
Great. And so this is essentially in one slide, Manitoba's clinical trial response to COVID-19. And some people might ask, why is a hematologist interested in COVID clinical trials? Well, the truth is, I'm not in particular, but I was privileged enough to have a team that was able to pivot quickly and start inpatient trials, because we did this for critical care and sepsis over and over again, where we were studying hematologic aspects of critical illness. And so early in the pandemic, in the spring, which feels like 8 years ago now, we mapped out the different touch points of COVID-19 patients, outpatients, inpatients on the ward, inpatients in the ICU. We didn't map out outpatient patients convalescing, which could be another and is another important aspect to clinical trials also. But this is what we thought we could handle early and with the resources that we had. And so we identified team members and trials pairing patients or people, healthcare workers, outpatients, with pre-exposure, post-exposure trials in patients with mild symptoms on the ward, with trials for that, like antivirals, anticoagulation on the ward, plasma, or really severely ill patients in the ICU, where we studied a host of interventions and continue to study those interventions. And our strategy is as presented on this slide. We embedded clinical trials into clinical care. And so whereby we have kind of in Manitoba had this really false and somewhat very much old culture in the province, where we separated research from clinical care, so how they're not connected in a sense. We embedded them, we paired them, because we didn't know how to treat these people. So every patient doesn't just inform our personal observation, but informs a national, international data set, so we can quickly arrive at an answer to figure out what works and what doesn't, adopt what works and abandon what's ineffective. We prioritized international trials as opposed to local efforts, despite their best intentions, because we really needed to get as many patients randomized quickly as possible to inform this current pandemic, not for the one that will occur in many years to come, hopefully. And the team is diverse. It includes a whole bunch of different specialists from the Department of Internal Medicine, also PhD clinical trialists. We have academic physicians like myself, Yoev, Sylvain, and clinical physicians like Glenn and ID physicians who aren't typically involved in leading clinical trials, senior, mid, junior faculty, medical students, a lot of project managers, coordinators. U of M is lockstep with us to shuttle contracts quickly. Issues of payment and ethics is working with us to make sure we can get these trials initiated and rolled out quickly. And even the government and the Manitoba has supported this initiative early, so that patients can have access to experimental medications under the right conditions. And this is like some of our team. We couldn't get everyone together for social distancing and scheduling reasons, but you can see several of them here. Brett Houston, Maggie from the ICU, Dana, Nicole, our sort of overall project manager, has been leading a lot of this. Multiple project managers who have joined our team, all on this slide here. So it's a really large team, probably about 30 of us in total. Next, we're going to talk about the evidence base. So how do we treat these people? We didn't have answers to any of these questions when we started short 6 months ago, and now we do have clear answers to several of them. To the ones that we don't have answers, we have ongoing trials. And so Sylvain and I are going to take you through some of these that are on the screen here. Okay, let me start with hydroxychloroquine. So this was our first go at this. And so when the pandemic hit, it was identified earlier, there's some in vitro data that suggests hydroxychloroquine could change viral replication by changing intracellular pH or affecting the viral replicative machinery. It was a repurposing drug that was available and inexpensive. And it was sort of my attempt to bring a trial here that could help flatten the curve. If we could reduce the severity of disease or reduce transmission to those who are exposed, well, maybe we could reduce community spread and also protect care workers, the healthcare workers. And so this was a trial that we brought to Canada, partnered with David from Michigan, the United States. And we just brought it and launched it to Canada. And we compared hydroxychloroquine to placebo for 5 days in patients who were, or people who were exposed, to see if we could reduce severity of illness, or their contacts, household contacts, to see if we can reduce transmission amongst the household. It was a really innovative trial. I had never done something like this before, and the team had never done it before. It was a totally internet-based trial where patients went to a website, enrolled themselves after learning more about the trial, participating in an online consent module, and then we shipped drug to them or placebo via courier, and all the endpoints were via survey electronically also to prevent spread of infection while participating in research. Lauren Kelly and Zena made a website within a weekend to house the trial, which is amazing. And then it turned out incredible. With a very small budget provided by the Manitoba Medical Services Foundation, we found this answer quickly with international collaboration with Todd Lee and colleagues from Quebec. And we published this in the New England Journal of Medicine, along with the key members of the study team, Lauren, Sylvain, Glenn, Nicole Martin, Lauren Kelly, just incredible team effort to achieve this, the results of this trial really quickly. And so what did we find? Well, we found that hydroxychloroquine didn't make a difference. There was no difference in new infection, no difference in hospitalization, no safety issues. And one could see this as a bust. It wasn't, it was a success because we believed in this early. There were physicians among us who were taking this with the thought that it would help and being pushed from leaders in our group without any data. And we provided this data that we should move on, and we did. And so, having no data isn't a problem for some, but it is a problem for us, and it should be a problem for us. This has been repeated since then. This was an impediment to the trial enrollment within Canada and the US, but despite the political influence, we persisted and found this answer.
I feel good about it. That's all it is, just a feeling. I'm a smart guy.
Testimonials is not a replacement for good science, and we can and will do better than this.
Thanks, Ryan. So with these politics and anecdotes, the FDA actually had approved this drug under the emergency use authorization. And several physicians, even here in Winnipeg, were prescribing themselves hydroxychloroquine to reduce their risk, as Ryan mentioned. Thankfully, we've quickly gathered trial evidence, not only in the post-exposure trials we were involved in, but really throughout the different settings and populations. And what you can see here from this summary slide of multiple trials, there's really no effect in any setting or population, and hydroxychloroquine has largely been abandoned. Similarly, a repurposed and mostly abandoned HIV medication, lopinavir ritonavir, or Kaletra as its trade name, had early hopes that it would be repurposed for COVID. Some local clinicians really advocated for use in our clinical algorithms. And a few months into the pandemic, good clinical trials started to surface that have demonstrated really no benefit to Kaletra, and it's since largely been abandoned. On the topic of abandoned drugs, remdesivir is another abandoned medication, an antiviral that was initially designed to treat Ebola and actually failed and was not a beneficial treatment in Ebola, and thus was repurposed for treatment in COVID. There remains controversy as to the extent to which remdesivir might help patients with COVID. So this is a breakdown of the evidence in the Act 1 trial, which was the first to be published in the New England in May. The authors demonstrated a significant reduction in the median time to recovery with remdesivir. So down from 15 days down to 10 days, saving you 5 days of symptoms. This was a very well done study, well conducted, randomized, double-blind trial, just over 1,000 patients, multi-centers, although most were in the US. But the benefit was largely seen only in the group on low-dose oxygen, and did not hold true in patients with critical illness on high flow oxygen or greater support. And notably, there was no benefit on mortality with remdesivir in the Act 1 trial. Since then, a much larger global solidarity trial that's run by the WHO has been released in preprint format that's demonstrated no difference in mortality or any of its secondary endpoints, including prevention of progression to mechanical ventilation or length of stay. If we go over to the next slide, when you pool all these trials together, there really is no mortality benefit, with a signal towards harm in the sicker patient population with critical illness. But in the lower risk group with a lower baseline risk of mortality, a non-statistically significant signal exists that there may be a tiny benefit in this group, which after more than 5,000 patients enrolled has not reached statistical significance. So despite the data, the FDA has approved remdesivir for use in all hospitalized patients. They did this prior, without factoring in the solidarity trial results into their analysis. And this is maybe because it's only out in preprint format, not yet peer reviewed. But this approval was perhaps rushed. And this is a trend that we've seen similar to other emergency use authorizations that have been released by the FDA for hydroxychloroquine, convalescent plasma, when treatments are so desperately needed. So any flaws in solidarity? Should we trust this trial? Why is the FDA and several clinicians jumping on board here? Well, many have criticized the solidarity trial, but this is a very well conducted, very generalizable study, which included more than 405 hospitals in many countries around the world. It was 3 times the size of the Act 1 trial. And while it was criticized for its open label design, this tends to bias towards observing a treatment effect. And there was no treatment effect seen. So this further supports the absence of a real effect there. Others criticized the inclusion of developing countries with poor standards for care, which may have washed out the effect. However, more than 50% of these patients were enrolled in developed countries like Canada and throughout Europe. And with more patients randomized from developed countries than the Act 1 trial put together. So I think the conclusions here can be that remdesivir doesn't work in the sickest patient population where we'd expect to see most gains, but it might have maybe slightly benefit at the earliest stages of disease in that subset of patients. So because of this, we continue to study this drug locally to further delineate its role. Despite the reluctance of many to study corticosteroids in viral pneumonia, probably because of a signal towards harm in influenza, which we see every year, steroids have been proven to be the real game changer here in the COVID-19 treatment story. This impressive trial, it's a platform trial called the Recovery Trial, demonstrated a 17% reduction in overall mortality for all hospitalized patients, and significantly improved other morbidity endpoints. In the subgroup analysis, which is shown at the bottom, the sickest patients on ventilators benefited the most, with an absolute risk reduction in mortality of 12.1% in the critically ill, which translates to a remarkable number needed to treat of 8 patients to save 1 life. A similar yet smaller effect was noted in the less sick patients who require oxygen therapy. But it's important to note that patients who do not require oxygen had a signal towards harm. and therefore dexamethasone is not being recommended for those without hypoxia. I want to emphasize the successes of the recovery trial. This is really a real life example of how a learning health system where research is integrated into care delivery can change the way we practice in very short order. Nobody knew how to treat this new disease in the UK several months ago, and most patients across the UK were enrolled in clinical trials. In some centers, more than 60% of hospitalized patients with COVID were enrolled in clinical trials to find the answers to these important questions. And this was number one, so that patients could be offered treatments that might actually work. And number two, so we could rapidly find out what works and what doesn't to inform the treatment of the next round of patients. So in 3 short months, they randomized more than 11,000 patients and answered the questions for 4 drugs, one of which, dexamethasone, is likely to save hundreds of thousands of lives going forward through this pandemic. So now we'll switch it over to Ryan to talk about the hematologic treatments.
Thank you, Sylvain. Yeah, I mean, I wanted to just echo comments about the recovery trial. It's like the envy of the world. They have randomized, that is the UK has randomized over 20,000 patients now. Recovery is just the ward-based trials, but within the ICU and the outpatient trials, over 20,000 patients. And they're about to report on convalescent plasma probably in the next few weeks to a month, I would think, because 1,500 patients have been randomized to that already in recovery, and then another 500 or so in the ICU group. And so it's really a model that countries will adopt after this, if they have any sense about them, in a sense, because it's just been so successful. Okay, it's great that Sylvain can wear the ID hat and present that data, and you did it wonderfully, Sylvain. I'm just going to talk briefly about the anticoagulation and hematologic aspects. And so it was recognized early in the pandemic that thrombosis and inflammation is a really prominent feature of COVID-19. And there was reports of like 5 to up to 30% of patients developing thrombosis, particularly in those who are most severely ill. And we know that these things are linked. And so there was all this eminence-based recommendations from people who were generally had their wits about them to like stick to the trials to sort of recommend intermediate dose or high dose. And then there was all this chaos of intracranial hemorrhage and other bleeding. And so we said, okay, well, let's come together again. When all the emotions settle down, we got to figure this out and organize a trial. And so there are many trials that are small, underfunded, and mostly likely to fail around the world, organized. But we do have 3 very large platforms enrolling into one single trial that I'll come back to, where we're going to answer this question: does therapeutic anticoagulation reduce mortality, progression, disease, and thrombosis in COVID-19? So this is coming. We're not sure what to do at the moment. The small trial has been published, 20 patients, has just been improved gas exchange, but we're on this right now, and over 1,000 patients have been enrolled. Convalescent plasma is a really important question to answer. Also, does conferring passive immunity to people with COVID-19 help them recover or prevent progression, disease, and mortality? Right now, the data the small amount of data that's emerged has not been completely encouraging. I have to say, Chinese trial, no benefit. Small trial, no benefit. This PLASA trial from India, almost 500 patients overall, showed no benefit. We are answering this really shortly from the UK, 1,500 patients enrolled so far, through a Canadian trial called Concours, and through that's slow to enroll because it's just national, not international, I might add. But it's getting there and picking up speed with our surge, and with some links internationally. And this international trial primarily in the ICU through BMAP. And why is this important? Well, here's a great example of why this is so important. And so early in the disease course, the US approved convalescent plasma for compassionate use. And after 100,000 patients were exposed to convalescent plasma, we still don't know the answer because it wasn't part of a trial. And I can guarantee you that if it just 5% of those patients would have been enrolled in a trial, we'd know already. And we'd know if these donors should keep donating, if the blood systems should keep processing, and if we should keep giving this to people because the benefits outweighs the risks. And now we don't know. This is the irresponsible part. We'll come back to. Sylvain.
So yeah, so coming off the dexamethasone story, the success of immunomodulators really appears to stop at corticosteroids. There's been much interest has been garnered about agents that block specific areas of the inflammatory cascade and different areas of the immune response. Interferon beta 1A is an example of that, but we now have robust data from another large Solidarity trial that really shows that there's no benefit. They're studying it as a nebulized medication, but it's not looking promising. Tocilizumab, which is another immunomodulator that blocks the IL-6 pathway, had really lots of interest early on in this disease, but small studies now are showing that it's probably not making a huge difference. There was some excitement about the Impacta trial in a press release that it might reduce the need for mechanical ventilation, but this has not been replicated across numerous amounts of studies. And again, the large platform trial Recovery is enrolling in one of its arms for tocilizumab, and this answer will be, we'll get this answer shortly. Anakinra, by the way, is another example of an IL-1 blocker, 4 trials in France, no benefit, most trials halted for safety concerns.
Days there, and I went in, I wasn't feeling so hot. And within a very short period of time, they gave me Regeneron. It's called Regeneron. And other things too, but I think this was the key. But they gave me Regeneron. And it was like unbelievable. I felt good immediately. I felt as good 3 days ago as I do now.
So this has been a recurring theme with the anecdote and what we're seeing broadcasted across lots of different channels. While there was a huge surge in interest in these monoclonal antibodies, both the Lilly product as well as the Regeneron product have recently released press releases halting their trials, mostly in the sicker patients that are hospitalized or with critical illness, showing either a lack of benefit or unfavorable risk-benefit profiles. So these have been halted.
Sorry.
So how do we put this all together? Really, I think the take-home message here is that we've been able to answer a lot of questions fairly rapidly in the span of a pandemic, but there really is no magic bullet for treating COVID-19, and most of these therapeutics, although they held promise and a lot of interest early on, and we were eager to start them, the science has shown that really most things don't work. So what we're left with is really excellent standard of care. Nothing special, nothing out of the ordinary, just oxygen, symptom control, DVT prophylaxis, IV fluids. We are recommending dexamethasone that should be given to all patients who are hypoxic. We're using a moderate dose, 6 milligrams. That's the dose that was studied. 6 milligrams once a day, that's equivalent to about 35 milligrams a day of prednisone. And we use that for 10 days, but we also stop it sooner if patients are well enough to go home. I think we have highlighted in the evidence story that the urge to treat patients with experimental treatment that we think must work, we should tame that response. And the safest and fastest way to get experimental treatments into patients is in the context of a clinical trial. Our COVID trials team is working hard day and night to offer patients different trials on the medicine wards. That includes therapeutic heparin, remdesivir, and convalescent plasma. And I think importantly, many of these patients are going to be really sick and anticipate a course where they may progress to critical illness. So clear goals of care planning upfront in their admission is important. There's really no easy threshold to use to call the ICU, but using your clinical judgment is likely to be most important here. Looking at the trends in oxygen requirement, the trajectory of your patient, and anticipating decompensations are likely a good start to get intensivists involved. If patients are requiring AGMPs on the ward, particularly high flow or non-invasive ventilation, it may be worthwhile to have discussions with the intensivists and infection control practitioners on the ward. Similarly, in the ICU, best practice is really just best standard of care. We need to use adequate but judicious fluid resuscitation, standard ventilation strategies, nothing fancy, using ARDSNet and prone ventilation when applicable. We do also administer dexamethasone, often IV up front, but oral is fine if the gut is working, and similar course for 10 days. And all other treatments should be considered in the context of clinical trials. Here in Manitoba, the REMAP-CAP platform is our workhorse in the ICU. we're enrolling patients in several different domains, namely heparin, convalescent plasma, and vitamin C. Just a quick note on antibiotic de-escalation and antibiotic stewardship. I think we are way overutilizing antibacterials for patients that are coming in with COVID-19. Many of these patients have a clear viral syndrome and a clear viral pneumonia with typical findings on chest x-rays. And we know that patients don't require antimicrobials for treatment of viruses. So unlike influenza, we know that bacterial superinfection is also rare in COVID, differing reports and observational studies, but for sure less than 5%. This is one that was presented at ID Week last week showing 3.5% superinfection. So I think it's important that we should avoid prescribing antibacterials in the right context, or we should de-escalate quickly when the clinical syndrome becomes more obvious. And unless there's really a sepsis syndrome or a clear bacterial pneumonia, we should reserve use of antimicrobials.
Thanks, Sylvain. In the last section, we wanted to highlight a couple of the trials that we're leading internationally and some reflections about the pandemic and some of the embedded clinical trials. And so, well, a lot of the trials, some of them we talked about, we are active participants with local leaders, which is hugely important. So we establish answers for ourselves and for the rest of the world. Manitoba is also leading international trials in a way that we haven't before. And I wanted to just touch on that briefly. And so in collaboration with two colleagues that I've never met in person, Patrick Lother and Ewan Gallagher from Toronto, we started a trial called ATTACK to study therapeutic anticoagulation in COVID-19. We originally enrolled patients just on the ward, and we did this in collaboration with national, international groups that are listed here: Canadian Critical Trials Group, CanVector, it's a thrombosis group, a cardiology group, international groups as well. It's actually now enrolling in 4 countries and 62 sites, and funded through various agencies. And so we started this, and then on the ward. And in addition to that, myself and Ewan started a similar trial in the ICU in an international platform called REMAP. That's studying therapeutic anticoagulation in Canada, US, UK, Ireland, Saudi Arabia, Australia, and New Zealand, and a couple others. In addition to that, we started a convalescent plasma trial in critical care with a colleague of mine, Alexis Turgeon. Subsequently, we've combined these trials. All these trials are platform trials, which is a type of trial design that we will see universally going forward after this. It's an enduring kind of trial that learns as it goes, where we might study something and keep studying it until a rule has been met. And the rule is either that the study stops for superiority or stops for futility, and we abandon the trial. If it stops for superiority, we adopt it as standard of care. We can add interventions as we go. So it's a trial that keeps on being relevant to study the next important question and dropping those that aren't important. With regard to ATTACK, the trial that I led on the ward, and REMAP, the one that I lead in the ICU, we merged these trials together. And one more merger come subsequently since then with the NIH in the United States, where now we have 3 platforms all enrolling under the same protocol. And so ATTACK is now extended to the ICU, REMAP extended to the ward, active both from the beginning. And we have one protocol amongst 3 platforms, all feeding data into one place in the United States. Our statistical consultant that's shared for pooled efficacy and safety. Actually, our first interim analysis is this Friday, I believe, with a common DSMB and common stopping rules. So the idea is to get an answer as quickly as possible and to adopt the therapy if it works or abandon if it doesn't. And so these are all very new methods. Like we call this the multi-platform RCT with a common or meta DSMB. Again, things that we hadn't really thought about before until we were challenged with the problem in front of us. So ATTACK now has 630 patients enrolled in 4 countries, REMAP 350, and active. It's starting to pick up within the United States, funded by the NIH, 50. So we have 1,000 patients that's going to be part of our interim this Friday. Okay, and briefly, some of the challenges. This is immensely difficult. We are all challenged with COVID-19 in many aspects of our life, no question. Focusing on some of the challenges of research that are relevant and just notable in discussions such as this. One is our culture. We have historically separated research from clinical care in healthcare, particularly in Manitoba. Not all centers do, by the way, now, and many are changing over. But it was a culture that was handed to us that endures, that is not appropriate anymore. What happens is that we have separate strategies where, we build research, we build clinical capacity to look after people like we're doing now, but we don't think about how we're going to care for those people and how we're going to get answers quickly if we don't have the answers. And so it becomes disconnected, where you have structures for clinical care and structures for research, thinking that they're not somehow linked, that the therapies we give right now, 50% of patients will be giving a drug for which may have an active component. And so what happens is that without these links to clinical care, we've got 100,000 people getting plasma in the United States without an answer. We've got health leaders, CEOs or CMOs, pushing to get a drug for compassionate use without really wondering if it works or not, instead of investing into research. And what if we did? What if we did have these linked? Well, we would have answers to questions that we don't already. And so I would submit to you, it's irresponsible for us at many levels to not know whether, a high volume paracentesis should be followed by albumin now. This is a question that internal medicine has been asking every resident for years and years and decades. But if every patient was just enrolled in a trial, we'd have this question answered 20 years ago. The best way to treat hyponatremia. Why don't we know this? If research was embedded in clinical care, we'd know this already. We'd know the answers for COVID already, and we'd know all these enduring questions that we've abdicated our roles to answer. Another challenge has been designing trials without all the knowledge that we need, and so we don't know the baseline event rates for a lot of outcomes, how effective treatments will be, and our typical trials design that are frequentist picks a sample size based on these things. And so we've had to adopt new trial designs called Bayesian methods that can function in the world of unknowns. And I think we'll see those enduring and being used routinely in trials post-COVID. We don't have a national strategy, and the UK has shamed us into understanding the need for one. And so a national strategy for research. So in the UK, they have 3 trials platforms approved, period. 3, 1 outpatient, 1 inpatient on the ward, and 1 inpatient in the ICU. And all questions are put through those through centers that were ready to activate, ready to activate with funded research infrastructure, using models that were filtered down instead of that were expanded up. And so that's the challenge with the existing funding models. Right now, we have a per project, basis where we compete for a grant, and so it's too slow. It's not responsive to the needs at the time. And what's gone on nationally, right? Like, so 3 trials or 3 efforts to fund an anticoagulation trial instead of 1, even though we were able to collaborate internationally around the 1. Funding that came out too late after other countries already got going with their clinical trials. And so the existing funding models are a challenge. Instead of funding teams or funding priorities based on established trial platforms. Politics got in the way, obviously, early in COVID, where edicts and testimonials were purveyed by leaders, some of which we highlighted, but not all of them for sure. We had issues with irresponsible publication of really poor quality observational data in high impact journals, and I'm sure the Lancet regrets some of this, and so does the New England Journal. But I think that's a lesson learned, is that it's just all this sensational poor quality data early that guides us, and then all of a sudden we have local leaders saying, Ryan, you must get hydroxychloroquine to everyone, you must get azithromycin, this will be a game changer. And of course it's all PAP. And we need to embed clinical trials into the way we care for people so we can arrive at the answer quickly, responsibly, to inform the current pandemic. And I mean, local bureaucracy, I live this on a daily basis. We all live it clinically and trials-wise, but we still don't have the Concord trial, that's a convalescent plasma trial going here, because it's taken months to get contracts signed through our existing channels of shared health or otherwise. And we really need to evaluate how much we value clinical research as part of clinical care to arrive at responsible decisions as quickly as we can, because right now they're not seen as complementary. And so facilitators are central funding, platform-based funding instead of project funding, which I hope we get to nationally and provincially. We can do this provincially too. We were able to lead a trial internationally because of startup funding through Research Manitoba, which is really a great feather in our provincial cap to provide startup funding for a team instead of a trial. Innovative trial designs that I mentioned a little bit, like platform trials, Bayesian trials, global collaboration instead of local efforts solely. And when we're done with this, we'll come out better. We'll use Bayesian adaptive platform trials routinely. We will probably not see the last of the multi-platform RCT. This experience has shown us that we can integrate junior faculty and trainees into the leadership of clinical trials locally and internationally. I might add, look at Sylvain, Lauren McKenzie, Lauren Kelly, etc. And we'll come out of this with increased capacity within the Department of Internal Medicine and University of Manitoba to design and conduct clinical trials. So in summary, yes, we're stuck with dexamethasone for the moment, and the best way we'll answer the enduring questions of remdesivir, convalescent plasma, and therapeutic anticoagulation, monoclonal antibodies, are to enroll each and every patient into a clinical trial. These trials will change over time as the next strategies become identified and incorporated, and as strategies get refuted for efficacy or for harm, or stop for efficacy or for futility, that is. And the novel designs will guide the clinical trials in COVID and beyond COVID. Okay, so I thank you very much for your attention. If you have COVID patients admitted, this is the clinical trials line, that's the number. So call us and we will, with our army of coordinators, really dedicated coordinators working almost around the clock with little resources, we'll get patients enrolled in trials so we can arrive at these answers quickly. These are the emails to Sylvain and I and our Twitter handles. This is like new for me, and Sylvain's helping me explore this. And so if you want to follow the progress of these trials, feel free. Okay, to ask questions again, go to Slido, put in the code there or press the link on the screen. We will handle some questions now as they come up. Sylvain, you've been monitoring this more than I will have been talking. So do you want to do the first question?
Sure, yeah. So there's just a comment from Dr. El Gabalawi here that a paper in Science showed 15% of COVID patients who were critically ill had autoantibodies to type 1 interferon before infection. So their plasma may actually make things worse. So another important comment that we don't really know what's in plasma. There's antibodies to other things, there's procoagulants, there's anticoagulants, and so important for us to study, whether or not these are beneficial or whether they're actually harmful. There's a few other questions. Maybe should we pass it to the panelists?
Yeah, so there was a question here. Maybe Yoav, it's talk about recovery. Enrolled pregnant women received prednisolone or hydroxychloroquine. Would you recommend the use of prednisolone or Dex for pregnant women with COVID-19?
It's an excellent question from Vanessa, and it's a tough one to answer. I don't know the number of patients, the pregnant patients in their recovery that were on prednisone and hydroxycortisone was small. And then you need to decide whether you want a steroid that crosses placenta. And so there's a debate about that, and with different recommendations, whether you use Dex versus prednisolone. And I guess if you expect that there's a risk of preterm delivery and in the window of 24 weeks and above, then maybe there should be a thought for dexamethasone, but it wasn't directly studied in the trial. So whatever I'm answering is kind of a made up answer. It's not based on the results of the trial.
Yeah, Anand or John, do you have anything in addition to that? Like, sounds like a reasonable advice to me.
Yep.
Sorry, nothing to add?
Yeah. Crystal Van Enneveld says some reports of older adults from observational study that vitamin D levels may impact outcome. Can you comment? Oh yeah, I'd love to comment on this. So any observational study of sick people shows that vitamin D is a problem. We have learned this over and over again, how the observational study has misguided single vitamin deficiencies. And all the randomized trials of vitamin D have been negative in my knowledge. So has any single vitamin deficiencies. These observational studies are of sick people with low vitamin levels to begin with. It's been proven to not be beneficial in, whether it's MI, multiple sclerosis, bone health, cardiovascular disease. The trials are just universally negative, and I do not think we should think that this is going to be particularly enlightening to study it in a randomized trial. Diabetes included, etc. They just go on and on.
Can I add to that? So again, I agree completely with Ryan, and the other. So we base it on plausible mechanisms and the effects of vitamin D on renin-angiotensin and the ACE receptor and on anti-inflammatory, and so always theoretical. But if we look at that in interleukin-1 and IL-6, so the fact that we want to believe that something has a theoretical leads us to bias in approaching it, and I agree that this is the low.
Thank you, Yoav. Vanessa says thank you too. The next, Charles asked a question about, have you made inroads into shared health and the need to rapidly turn around research paperwork? I mean, I think everyone's trying their best. I think that there was a lot of turnover in shared health when Eric and the leadership left. Paul Bowden's taken over. Legal expertise has changed. I think that the culture of separating research from clinical and not seeing the benefit of integrating those has hurt us and the organization as well. I do think that radical changes are needed. We need to work with partners to launch trials as quickly as we can. It's been a struggle to do that, and we can do better. And I hope that we will come together as a team, as we always have in Manitoba, to do that, to do better. How confident are you that using Bayesian methods for stopping early will not lead to false positives? Certainly a challenge. Yeah. Do you want me to tackle that? I can turn it over to anyone as well. This is a challenge. What's the alternative is the question. And so take, ATTACK or Concor attack anticoagulation or therapeutic plasma Concor, where we not even sure the baseline and we guessed at the beginning for Concor. Rates of hospitalization and intubation, which has now changed as we've gotten more into a groove and adopted evidence-based practices. We've introduced dexamethasone and it's reduced mortality, so the baseline outcomes have changed. We didn't know how effective the treatment would be in whom. And instead of having a trial learn as it goes, looking at the data as it evolves with really conservative stopping measures, we guessed. We flipped a coin. We didn't flip a coin at a patient level, we flipped a coin at a trial level, which will be wildly overpowered or wildly underpowered, and I suspect underpowered. And so the alternative of learning as you go with the Bayesian method and using conservative strategies to stop when you're certain, like 99% probability of the treatment being effective, is wholly more appropriate than that alternative in my view and the view of many around the world now. And so I think that there's benefit. I think there's conceptually benefit, but I agree that it's more complicated, but we have a lot of experts involved to help us guide those methods as well, for sure.
I can answer one of the next questions here that's coming from someone, CB. Why should Dex be better than or different than prednisone or hydrocortisone? I think that's an important point that we didn't emphasize too much in the presentation. Dexamethasone was chosen by the recovery group mostly because of the lack of mineralocorticoid activity. So in theory, less salt and water retention and less effects on pulmonary edema. So that's the theoretical benefit to using dexamethasone. Interestingly, the dose that they chose was also a very moderate kind of middle of the line dose, because probably too high of a dose could be harmful, which is what we saw with influenza for the most part. In theory, it shouldn't matter which steroid you use. And in fact, there have been other studies looking at hydrocortisone and prednisone, and it seems that it doesn't matter. The mortality signal is there, the mortality benefit is there regardless of the molecule that you use. So if, in the absence of dexamethasone, it wouldn't be wrong to use hydrocortisone or prednisone at equivalent doses.
There's a question on ARBs with regard to the viral dynamics, adhesion via the viral spike protein and angiotensinogen receptor blocker help mediate or reduce infection likelihood. Yeah, for sure, this is being studied. And so we have an ARB domain as part of the REMAP. So REMAP is the randomized embedded multifactorial adaptive platform trial that therapeutic anticoagulation and convalescent plasma is built into. And there is a domain studying ARBs that is, I don't think it's launched yet, but it's certainly finalized and will likely be launching soon. How many trials can a patient be on? And so I'll take that if people don't mind, is that almost all the trials except for the industry trials, and in fact some industry trials too, have adopted a strategy of widespread co-enrollment. We look for interactions, a priori and post trial. But if you can imagine a clinical practice that might have lots of people receive different co-interventions. The more interventions there are in a trial, actually, the more it likely reflects clinical practice, to be honest, and individual decision-making. And so co-enrollment has been widely adopted in all the trials around the world. And so patients can be enrolled in multiple trials. And to that extent, consent has been a challenge. And so Manitoba has pioneered a new method of consent with the help of our REB and Lauren Kelly, Brendan Foote, Nicole Martin, and many others who contributed to a harmonized consent form where patients are approached once with front matter and trial specific matter to understand all the trials going on at once and what they're eligible for and what they want to participate in.
Just to add to that, in terms of the REB, to answer some of Charles Bernstein's questions, so there are some bureaucracies, but to add to what Ryan said, I think we need to give kudos to the REB that has been pretty open-minded and through most of it rapid and open-minded to new platforms and looking at new platforms that have supported us through those studies.
There's a question about the Toronto group is showing some benefit with lambda interferon, or we're looking at it locally. I can take this, but others can as well. I don't want to hog this at all.
An end here, I can do that. Please. So interferons have been broadly shown in vitro to have antiviral activity. And at different times, people have looked at interferon alpha, beta, gamma, now lambda. I'm not aware of any work locally looking at that. There was a trial that came out recently, relatively small, I think. They're certainly not huge, that demonstrated, I believe, that interferon alpha didn't seem to provide any benefit. I'm not aware of any other major trials looking at interferons that have produced any results yet, although I'm quite certain that there are several other large trials.
I mean, interferon took a bit of a hit with the recovery and the United States trial, and stopped now in REMAP and World Solidarity for harm. And so this will be approached cautiously going forward for sure.
Yeah, having said that, the different interferons seem to have significantly different activities. So I think it'll continue to be a question.
Yeah.
I was hoping I could just ask John a quick question here about the management of patients on OptiFlow on the ward, and whether there are concerns about this being aerosol generating, and whether or not the ward is equipped from an infection control perspective to handle patients on OptiFlow.
So you're asking a nearly impossible question to answer. We're doing the best we can with what we've got. If we were dealing with a facility or facilities with private rooms, these discussions wouldn't even exist. Regrettably, we don't have those facilities. So are the facilities that we've got capable of handling patients with OptiFlow? Of course they are. We just have to use common sense and wear our PPE.
There's a question about ongoing RCTs on PPE or other protective trials. And actually, I have been only following the therapeutics closely. And so I would defer to my colleagues for this.
I'm not aware of RCTs. Of course, there are some studies that we're, doing locally on, decontamination, sterilization of N95 masks and reuse and such. But I'm not aware of any randomized trials on this subject anywhere. John, do you have any insight on that?
Yes, Mark Loeb from Hamilton is actually looking at N95 versus procedure mask. However, I'm not sure where that trial is. We were asked to participate, but for a number of logistical issues, it wasn't appropriate for us to engage in that study.
Okay, well, that takes us to the top of the hour. I wanted to thank Yoav and Nan and John for joining us to help answer questions. It's great to have your expertise, and wonderful to see Sylvain taking such a leadership role in the trials. This is his time, not just to shine, but to get great experience as an ID intensivist, really getting his hands dirty in trials, learning the ins and outs of clinical research embedded into clinical care. He's doing an amazing job, and he's going to be a star in Manitoba. It's really a privilege to work with you and help supervise you, Sylvain. And if anyone has other questions, email Sylvain or I. And again, enroll these patients into trials, help us get the answers as quickly as possible. And we'll provide updates as data becomes available.
Thanks so much. Have a great day, everybody.
Thanks
very much.